Prostate cancer is the third most common cancer death in Australia in 2014. Prostate cancer tumors are often removed as a part of standard treatment, but residual or secondary tumors are known to be dormant for an extended period of time before recurrence. We hope to prevent cells from developing the tumors by impairing their ability to sustain proliferation regardless of regulating signals. Generally, we find an overexpression of androgen receptor within the tumor cell that allows them to be self-sufficient. One possible target that has been identified is retinoic acid receptor-related orphan receptor gamma. ROR gamma is a regulator that enhances the expression of androgen receptor within the prostate cancer cells. We hope to inhibit the action of ROR gamma in order to restrict downstream cell proliferation. ROR gamma directly binds to the promoter region of androgen receptor via the ARROR response element. Through the recruitment of nuclear coactivator 1 and 3 activating gene transcription and expression of androgen receptor, this allows 5-alpha dihydrotestosterone to bind to the androgen receptor, which undergoes conformational changes and leads to the release of heat shock proteins. This results in interaction between the N and C terminal of the androgen receptor, allowing the binding of important alpha and translocation of AR into nucleus, where dimerization occurs. The receptor dimers bind to the promoter region of the target genes, such as prostate-specific antigen and transmembrane protease serine 2, via androgen response element, resulting in the transcription of the target genes and enhance the proliferation and survival of prostate tumor cells.
Our drug, by suppressing androgen receptors at the transcriptional level, may therefore offer a therapeutic advantage over current anti-androgen therapy. Furthermore, the antagonists show greater and long-lasting reductions in tumor size compared to enzalutamide without overt toxicity in mice. Likely because of the undetectable level of raw gamma in normal tissue, the antagonistic effects were tumor-specific. However, suppression of androgen receptor expression may lead to an elevated androgen level, hence resulting in infertility. During androgen therapy, the cells can develop a resistance to treatment, where the pro-apoptotic signals aren't received by the prostate tumor cells as a result of AR activation. Our antagonist is capable of preventing the production of AR in specifically prostate cancer cells, hence enabling the bypass of AR mutation resistance and ligand-independent AR activation resistance. However, if the transcription and synthesis of the AR pathway itself is mutated, the raw gamma antagonist may be ineffective. A way to keep the immune system from recognizing the antagonist is by using pegylation. PEG stands for polyethylene glycol, and this acts as a shield by evading the immune system entirely. Bifunctional PEG chains are used to bind onto the drug on one end, leading it to the target site to react with the cell, and a targeting agent on the other side. After use, it biodegrades as they are made up of liposomes. It is able to improve the pharmacokinetic and pharmacodynamic ability of a drug. For example, pegylation helps to increase the drug's half-life and promote the enhanced permeation retention effect, causing preferential accumulation of the drug in the tumor cells rather than the anti-tumor cells. Though PEG increases half-life, it can also cause toxicological effects from its bioaccumulation of large molecules in the liver from the variable time clearance. Raw gamma antagonists were recently tested on xenografts generated from prostate cancer cell lines. A suspension of prostate cancer cells, PBS, and matrigel was subcutaneously implanted into both dorsal flanks of severe combined immunodeficient mice. Once the tumor growth reached approximately 80 mm cubed, the mice were treated with raw gamma antagonists. Tumor size was then measured with calipers. Raw gamma antagonists were also tested on the growth of orthotopic prostate tumors in SCID mice and found to be effective in preventing further tumor growth. With more funding, we are hoping to advance to the next stage and insert orthotopic xenografts into humanized mice rather than immunocompromised mice to better mimic the environment of natural prostate cancer tumors. In the future, we hope to investigate how our raw gamma antagonists would interact within an environment that mimics the human immune system using humanized mice. Although in theory, pegylation would allow us to bypass the immune system, we need to have solid evidence of it working with our raw gamma antagonists before moving to human subjects and clinical trials. We would also like to gather information on the clearance rate of different antagonists, but that would mean longer treatment periods to observe any adverse effects.